Okay, hello, my 365 class, plant physiology. So let's try to do this home-based experiment on seed germination. It really shouldn't be too complicated because you have everything at home, so you should have all the material that is needed to do this experiment. So it will be completely home-based, and you have a lot of times and all the materials, so this is, I think, perfect to make up for the labs we didn't do. You can choose any seeds that germinate fast, such as beans, peas, radish, pumpkin, squash. All of those seeds we germinate within the couple of days. Usually, it takes them a week or two to germinate. So you will be recording the germination rate. So you will actually count the number of seeds that germinated. So that is all you need. Now, there's several ways you can design your experiment. You can try putting the seeds in the soil. So I will talk about that later. Or you can try a paper towel method. Now, this method is used to kind of check whether the seeds are viable, meaning that they're not dead. So here is a YouTube video explaining how to do that, but basically you just pre-moisture the paper towel, spray a little bit of water, then you lay 10 seeds evenly spaced on both halves, and then you put all of that in put it in a Ziploc bag, and then in a warm, dark place for a couple of days, and then check how many seeds have germinated. So that is the easiest way. So that can be your control, and then you can have the same setup with spraying the seeds with any type of other solution that you are experimenting on. Other options is that you can use filter paper or sponge, which is placed in a jar. Or if you decide to use soil, what you have to pay attention to is that you have to put the same amount of soil in all the cups. So make sure you have the measure in grams. And I really like these cups, which are transparent. If seeds germinate in here very quickly, so make sure you use all the same cups. So pretty much light conditions, amount of soil, amount of water, all those variables should be bring to a minimum. So really we are only trying to separate the control group and the experimental group. So what can be your treatment? Of course, you will have the control group, which you will water with normal tap water. I'm not sure that you have access to distilled water, so you can just use a normal tap water. You can try different salt concentrations, sugar concentration. You can try soap, detergent, any treatment that comes to your mind. This is a really freely experiment, so any treatment that you think would be interesting to test, you can use. Always bear in mind to have the same conditions for the control and the experimental seeds. Now, the seeds that you obtain will have different genetic structures, so that might alter your results because these are not hybrid seeds that you will obtain. These will be, genetically diverse seeds, so, make sure to mention that in your report. Okay, so that is that. Now, this is the report that you have to fill in. So you gotta state the problem. So what is the effect on whatever treatment you're going to use on the germination on whatever seeds you choose. So a little bit put here information about your seeds, how they germinate, what are the requirements, and so forth. State the hypothesis. Then talk about, write here a little bit about the experiment, such as materials, what are dependent and independent variables. Write down the procedure. And of course, in the end, you will record the seed germination rate. After you're done, you have to start collecting the data. So, let's say 7 days. If your seeds haven't germinated within the 7 days, then you have to expand this column and give them another 7 days to germinate. So it really depends on the type of seeds that you choose. So in this table 1, you will fill in for the control group. So you have total seeds in treatment. This again will depend on the number of your seeds, because if you read, for example, usually beans are placed and they germinate best when they're placed in the numbers of 3. So it really depends on what type of seed you choose. So do a research, and then it can be 1 to 10 seeds in the treatment. So of course, starting from day 1, there will be 0 seeds germinated, and then as the days progress, you will count the number of seeds that have been germinated. So then you sum all of those up in this table, and then you calculate the percentage as well as cumulative percentage. And how you do that, this is the explanation. So you need to divide that time with the total seeds in treatment. So of course, this will depend on how many seeds you put initially. For the table 2, you record the same data for the experiment. Now, it doesn't have to be milliliters. You can put, if you're watering this one, you can put grams of maybe some fertilizer or anything. So this really depends on what type of experiment you are designing. And again, you are recording the number of seeds that have germinated. Here you're going to put a graph that will best represent germination rate. So you should have 2 lines, one for control and one for experiment. Then you will have to do a t-test. Now, in order for you to do a t-test, you need to have at least 5 repetitions. So you kind of need to have 5 controls and 5 treatments. So you need to repeat the same experiment and at least 5 times so that you can do a t-test. You can set up everything all at once, or you can choose to experiment over 5 weeks, but I don't think we have that enough time. So you can set up the experiment just once and then do the t-test. For this table, I have given you an Excel table, so you will put the number of seeds germinated here in the control group from day 7. So in here, it's the number of seeds that have germinated on the day 7 or on the day 14th, depending on whether you have some seeds germinated or on day 7 or not. And then you also fill that up for the experiment group. And I have put here a formula which you are going to use. So once you fill in this column and once you fill in this column, then you are going to get a number. So if the number is larger than 0.05, then there is no statistical difference, meaning that if you treated the seeds with whatever fertilizer, it makes no difference. And if the number is smaller than 0.05, then there is a statistical difference. Okay, then this means that your treatment is working and there is statistical difference. Okay, so all of these files I'm going to post to make things easier for you. So after you've done that and do the repetitions, you have to write down whether there is a statistical difference between the two groups and why. And then in the conclusion, you have to write down whether your hypothesis was supported or was not supported. So please elaborate on that. And number 7, in all research, rather it's a small scale like this one or a big scale, we want other researchers to be able to repeat your experiment. So, kind of write down a statement that you design your experiment to, make the variables less variable as possible, and make sure the seeds were placed in the same amount of light and temperature and so forth. So fill that in as well. So this is what I will be looking from you. Oh, of course, and then here in the end, please put some images. Okay, so images will go right here. Take photos with your phone and then insert a couple of images right here. Okay, if you guys have any questions, please write me an email and then I will be happy to reply. The deadline for this is 15th of April. So by evening time, 15th of April is when is the deadline. So you have how many weeks up until that? 1, 2, 3. So almost 4 weeks to complete this experiment, which is enough time to do that. Okay, so the deadline is the 15th of April. We don't know at this point when the UB will be open. If this virus, takes hold, they might, postpone finals, prolong school. So who knows what is going to happen? But I still have to give you a deadline. Okay. If there are any questions, please feel free to either email me or send me a WhatsApp message. And like I said, I will be happy to reply.